We talked about how to separate proteins. Now we're going to talk about how to identify them. The most common way of identifying the presence and quantity of a protein is a technique called the ELISA technique. ELISA stands for Enzyme-linked immunosorbent assay, and this is a common way in the biochemistry laboratory to see whether you have a particular protein in a sample and how much you have. It's also sort of the basis for a majority of the hospital laboratory tests that detect proteins. There's some sort of ELISA variation or ELISA itself. Also, you find a bunch of ELISA tests for home use assays, stuff you would pick up in Walgreens or CVS to detect particular diseases. Now, in order to understand what ELISA means, we have to understand what this immuno part means. We're going to use antibodies as part of this test, and therefore we have to understand a little bit about the biochemistry of antibodies in order to figure out how we're going to make them and how we're going to use them to detect a protein we're interested in. So let's watch part of a video that talks about antibody structure, and we won't watch the whole thing, but then we'll move on to see how we're going to use these as in our ELISA.
Antibodies are made of several different peptide chains, bound together in a Y shape. An antibody is made of two identical immunoglobulin heavy chains and two identical immunoglobulin light chains. Roughly speaking, the structural features of an antibody can be split into two parts: the domains that recognize a target, known as the variable region, and the domains that determine the antibody's biological function, known as the constant region.
Okay, I'm going to skip ahead a little bit so we can learn about the
heavy chain
region.
An antibody binds its epitope through the variable regions in the tips of the heavy and light chains.
This is the antigen.
There is an enormous amount of diversity in the variable regions, so that different antibodies could recognize many different target epitopes. Each of these chains contains three complementarity determining regions, or CDRs, located at the tips of each variable domain. Parts leading up to those tips are called the framework region. Most of the diversity between different antibodies is generated within the CDRs. Together, the three CDRs form the epitope binding site and determine the specificity of individual antibodies.
Okay, so in antibodies we have constant region and variable region, and it's the variable region which is responsible for binding to an epitope. Right? An epitope is just really the definition of an epitope is something that is recognized by an antibody. Now, if we're trying to detect, let's say, this green protein here, there are regions on that green protein that have the potential to be recognized by an antibody. So how do we get an antibody that recognizes an epitope on the protein that we're interested in? Well, let's imagine that this green protein is an Ebola virus protein, and we're interested in detecting this Ebola virus protein because we want to have a clinical assay for whether someone has contracted Ebola or not. Now, this is a viral protein. It's coded for by the viral genome, and so it can be isolated. The gene for it can be isolated. We can make some of that protein in bacteria and purify it using chromatography, and we can have a sample of this purified Ebola protein. Now, that Ebola protein can then be injected into the blood supply of any animal that has an immune system, and that immune system will recognize this viral protein as not normal, not part of that animal's normal protein complement. And so the immune system will react against it. This will act as an antigen, and there'll be an immune reaction against it. A very common animal to do this with are rabbits or mice or sometimes even goats. Rabbits are the most common. So we'll take our Ebola protein and inject it into a rabbit. The rabbit will have an immune response against it, and generate antibodies against it. Now, these antibodies will have variable regions that can recognize various epitopes, probably all over this viral Ebola protein. Now, those antibodies can be harvested from the rabbit, from its blood. We isolate the serum from the rabbit, and we have polyclonal antibody preparation. Now, it's polyclonal because there's probably lots of different antibodies that bind to lots of different places on this Ebola protein. One antibody might bind here, one might bind there, one might bind there. We're just taking the whole serum from the rabbit. So any antibodies that bind to it are going to be in there. There are other types of antibody preparations called monoclonal antibodies, and we might talk a little bit about how you make those in class. Okay, so now we have an antibody that can bind to the Ebola protein, and that antibody is a rabbit antibody. And so the constant region of that antibody is really distinctive for a certain class of rabbit antibodies. So what we're going to do is we're going to take a bunch of patients, and we're going to take their blood, where we think the Ebola protein might be if they're infected, and we're going to spot it onto a membrane, piece of filter paper or something, so that that patient sample stays in place. And then we're going to treat those patient samples with what we're going to call the primary antibody, the rabbit antibody that can react against Ebola protein. Now, if a patient has the Ebola protein, when that primary antibody interacts with the piece of paper, it will find the Ebola protein and the variable region will bind to it. Okay. We'll then wash away all unbound primary antibody, and we'll treat this piece membrane with what we call a secondary antibody. Now, the secondary antibody comes from a different species of animal, maybe a goat. And this antibody has been generated to recognize the constant region of any rabbit antibody. So the constant region of the rabbit antibody is acting as an epitope or the antigen for our secondary antibody from a goat. So the goat antibody will come in, bind to the primary antibody, and it will be conjugated to an enzyme. Okay. So what we've done is, if on our patient sample there's the protein that we're trying to detect, it will catch the primary antibody will catch the secondary antibody, and the secondary antibody will bring along this enzyme. And the enzyme is really the key to detect this complex here. Now, the enzyme's a catalyst. It's going to catalyze a particular reaction, so we put the substrate or the reactant for that reagent in there. If that enzyme's there, the enzyme will catalyze the production of this product, and the product is something we can see. Maybe the product's blue or red, maybe it gives off light, maybe it gives off fluorescence, something that we can detect. And so then we'll analyze our patient samples, see which ones light up or change color. And the only way they would have changed color is if the enzyme's there. The only reason the enzyme's there is because the secondary antibody's there. The only reason the secondary antibody's there is because the primary antibody's there. The only reason the primary antibody's there is because it's holding on to the protein that you're interested in. Okay. So those are ELISA tests, right? And they rely on antibodies, and antibodies have great specificity, so you can engineer antibodies to detect almost anything you're interested in. And using this ELISA technique, you can detect the formation of this complex and the presence and quantity of how much of your protein of interest is present. Pregnancy tests use a variation of the ELISA. These are ones that you've probably seen in the drugstore. They detect a protein called HCG. HCG stands for human chorionic gonadotropin. It has quaternary structure. It actually is a dimer, a heterodimer, with an alpha subunit and a beta subunit. And it's used as a good marker for pregnancy because after gestation, the levels of HCG which here are in these squares, go up really quickly and to high levels. And so it's a good marker to detect pregnancy pretty early. So how do these pregnancy tests work? Well, what we've got, let me change the color here, is a test strip, right? And HCG is present in the urine. And so a patient will pee on one end of this strip, and if there's HCG in there, it will have a ELISA-like reaction occurring. So let's look and see what happens. Okay, if there's HCG in the patient sample, in the alpha and beta subunit will bind to an antibody present in the strip. Now, this antibody binds to the beta subunit of HCG and usually has some sort of colored bead on the end of it. Now, you can't see that colored bead because these antibodies are sort of diluted all over this test strip. But if there's HCG, they're going to hang on to the beta subunit. Now, as the sample, the urine sort of travels down this through capillary action, this filter paper in the test strip, they're going to encounter first the test area. The test area has another antibody that binds to the alpha subunit of HCG. So let's watch this go down. Boom. If there's HCG present, the test strip antibodies are going to catch that alpha subunit and really concentrate these colored beads so that you might see a blue colored line on the test strip. Now, there's plenty of these first antibodies in the test strip, so some of them won't be bound to HCG. There's just not enough HCG for all of them, and they'll continue down the test strip, and they'll bind to a third antibody that's present in the control area of the test. Now, again, they'll be caught by this control area, and these beads will accumulate, and you'll see a blue strip here. This is just an indication that the test worked, right? If these antibodies with the beads made it all the way down to the control, then they had a chance of being caught by the test. So it makes you feel good about a negative result. You just didn't pee in the wrong place or something. So if you see two lines, beads accumulating here, beads accumulating here, then that's a positive result for pregnancy. If you only see a blue line on the control, that's a negative result. If you don't see either line, it means that something screwed up with the test. Right? The only reason you get a blue line here is if you had HCG present, which sort of builds this complex between these two antibodies, allowing the accumulation of the color.